Okay, I think we're just about ready to go. So, yes, we'll take your question, please.
So I've got what should be a simple one. It's with regards to doing intranasal delivery. Has anyone found a good way to do local exposure with a bioanalytical method in the nasal cavity as opposed to just doing systemic levels as a part of any of these studies?
I'll take that one to start with at least. So yeah, quite often you're talking about low amounts delivered, rapidly cleared, and not really seeing a lot systemically. I have seen some people actually take the nasal tissue, homogenize it, and analyze that. That's one way around it. Another way around it I've seen is to use a surrogate biomarker of efficacy to demonstrate exposure rather than trying to analyze for the drug itself if it's impossible to do so, if your method isn't sensitive enough, or if you just, you can't track the drug itself. So those are two things that I've seen done. Another thing that I've discussed with clients as well is for clinical studies, they sometimes use nasal strips. I haven't personally done that yet, but it's something that we might need to consider.
I was actually going to go a step further, and if you haven't done it, then you won't know the answer. But has anyone seen those actually work? Would be my next question.
Not yet.
Okay. Thank you.
Okay.
All right, I've got another question that I think might be simple. It's to do with the bolus administration by human products versus the prolonged exposure time. I'm just curious to hear what's the kind of current understanding of to which extent we sort of the current framework picks up the risks for kind of bronchospasm or triggering cough, which might be sort of triggered by the amount of drug that is presented at a single point in time. So is there any kind of what's the thinking of the evidence that the tenfold factor or fourfold factor is sufficient to have a safety cover for that?
Who wants to take that?
I can maybe answer to that, but well, actually, rats don't cough at all, so you wouldn't pick up anything in a rodent model for those particular risks. You might pick up evidence of bronchoconstriction in a non-rodent if you see changes in plethysmography, but sometimes, and you might pick up irritation in your pathology, which would suggest that potentially when you go into the clinic, you might see signs of irritation. So if you start to see that in your upper airways or something, you might predict that the clinical outcomes would be that coughing could be one of the consequences, and you would have to build the monitoring in. Typically in inhaled clinical trials, you'll have the FEV1 spirometry measurements, and you will pick up any adverse events of coughing. I mean, you hope not to go into your trial and have all these issues of irritation, but you should get a good indicator from the pathology and the clinical signs in the non-rodents and any respiratory plethysmography data.
Yeah, you certainly can see coughing in dogs if there's sufficient irritation of, the larynx and the vocal cords and so on. We've seen that definitely.
Yeah, thanks. That's clear. It's monitorable in the clinic.
And it would always be monitored for, yes, absolutely, in clinical trials. So, if you see it in your animal species but you don't see it in humans, then that's okay. But I take your point about, we're not giving bolus inhaled doses to animals because we're having to achieve those safety margins that are required by the regulatory authorities. And, that may be somewhat arbitrary, possibly, but it's what we're working with currently.
I was curious to learn a little bit more about why mini pigs became the preferred non-rodent model, and how its physiology might compare to, say, dogs.
I think from our perspective, it's just the the public perception of the use of dogs versus mini pigs. But I must admit, I've not seen personally the background for the decision.
Yeah, is
How
it,
about the physiology comparisons?
Yeah, I think from my perspective, there are some suppliers, let's say, of animals out there that push the use of the mini pig over the dog. But I mean, I would go back to my earlier comment that I think, and maybe Tony can back me up here, is that mini pig studies by inhalation, you could probably only go out to about 13 weeks, maybe 4 weeks, maybe 13 weeks. So if you have a chronic indication, then I would just stick with the dog.
Yeah, I certainly agree with that. I think, as well as the fact they just gain weight so quickly, so the whole dose thing becomes difficult. The AIT equation that we use for the minute volume becomes increasingly inaccurate because they're just getting fat. Their lungs aren't growing. So yeah, I share Paul's view that the dog probably is a better model, but at the moment we're stuck with this UK Home Office decision that you should use the mini pig in the first
So are they
instance.
truly mini pigs, or are they just young pigs?
They're smaller than traditional pigs.
They're not domestic pigs.
No. So they're about 10 kilos for a short study. By the time you get to a 4-week study, they tend to average 14 kilos over those 4 weeks, but they start at 10 and they get to nearer 20 and you get an average of 14. Beyond that, as Paul says, that they're just going to get, they put on so much weight so quickly.
I mean, the other thing that we end up using as a justification quite often for not using the mini pig for inhaled studies is that there isn't a lot of historical control data for the histology. And I think, as we've heard from a lot of the presentations today, or certainly in my experience, it's the histology locally in the respiratory tract that usually drives the no adverse effect level in a study, and that's what you ultimately use to define your starting dose in the clinic. So if you can't be confident in being able to interpret the histology, then that's not the right species to choose.
Okay.
this side?
Is there a question?
Yeah. So the importance of trying to optimize the aerosol particle size distribution for different species came up once or twice in the presentations, and in my experience, certainly for dry powder formulations, this isn't really done. You make what you make with the intended purpose of dosing it to humans eventually, and that just ends up getting used in the toxicology assessment. So how important do you think that really is?
I think for me, we would always target 1 to 3 microns for rodents. We would request that the material is micronized. Most often or not, for pharmaceuticals, it is. Yeah. So we can achieve that target of 1 to 3 microns. But obviously, within the 1 to 3 microns, you could have a particle with an MMAD of 1 micron or towards the upper end 3 microns, and that could result in an important difference as well. So we don't look so much at that granularity of the particle size or the MMAD within that 1 to 3 micron range. It's just the accepted range for those species.
Yeah. I mean, I think luckily the formulations often fit within that range anyway. But another thing I've experienced is I'm sure other people would say the same thing, is when it comes to the running of 14 or 28-day toxicology studies, we'll often be going, how many kilos do you want?
Yeah.
So I'm wondering if more effort was made to reduce the particle size distribution to suit the species, whether you would get more in effect, and therefore actually the material requirements overall might go down. Is that a strategy?
I think from my perspective, it's the greatest amount of compound is required because of the losses within the inhalation system, as opposed to refinement of the particle size. So, I mean, there are, I guess, certain ways that we can try to improve the system efficiency and to bring down your compound requirements. But in many respects, we are limited with the equipment that we have. I mean, so for example, in non-rodents, so primates or dogs, the aerosol is generated into a conditioning chamber and then conducted to the animals just by flexible tubing. And that needs to be about 1 meter in length in order to deliver it to each animal within the group around the central aerosol plenum. So those losses are quite considerable, and it's difficult to try and avoid that, to be honest.
Okay, thanks.
I mean, just to add to Paul's point about you would request the material to be micronized if when you first did the trials, you were getting nowhere near the required range. And certainly I wouldn't give the go-ahead for a study to start if the particle size measured in the chambers wasn't appropriate for the animal species. You just wouldn't do the study, as there's no point. But there are other methods you can use to sort of get rid of the bigger particles. Again, that's wasteful, obviously, but obviously micronization is the way to go initially, really.
Yeah, and I think the other thing I would add is that obviously you need to give the animals a good supply of air so to meet their demands. So, and you need that safety factor in there as well. So you can't take risks with the animal welfare. So.
Okay, I can't see if there's any more questions. I'll ask in the meantime, Joe, you were talking about your NAMS work stream. Can you just elaborate for me? I think I might know, but maybe not. What are NAMS?
New assessment methods. So really we're talking about some of the really nice models that Vicky was talking about. So one of the questions, I mean, as I said, I started life as doing in vitro work and in vitro and vivo correlations. So that's, some decades ago now, and we haven't quite got to a stage where, certainly in the pharmaceutical sector, that these sorts of tests are accepted as a true alternative to in vivo studies. We still have to do the in vivo studies that Paul talked about as a regulatory package of studies. I think where we're making some headway now is perhaps in the ag chem area more so, where there was a really nice example that Vicky alluded to on her slide, the AARTA thing with chlorothalonil, one of the, and it was a Bravo 720 formulation. So, I mean, when I was a study director working at Central Tox Laboratory for Syngenta, I actually did some of those acute inhalation studies with chlorothalonil, and it's horrible. We had to literally put Vaseline on the faces of the rats because they just reacted. It's so irritant. Their eyes swelled up and everything. So it's not a nice material to work with. But because of the effects that you see in the shorter term studies, you would then be required under ag chem regulations to do a 90-day study with that material. And you just can't do it. So this example was really nice where they used I think they'd done maybe up to a 28-day study as an in vivo study already, recognized that although they should do a 90-day study, they were never going to be able to do it because of the irritation. But then looked at some of these other methods that Vicky described, the alley culture, some modeling as well. So computational fluid dynamic modeling to generate some realistic exposure data. So this is really about occupational or accidental bystander exposure data and a risk assessment in that setting. And by putting all of those things together, they were able to derive margins of exposure, which allowed them to not have to do that 90-day study, which ordinarily would have been required. So that's the only example I know of where you can actually say that these new assessment methods have replaced what would have been a 90-day in vivo study. But in terms of for pharmaceuticals, we're not there yet. I have seen almost all of those methods that you talked about used in packages. Some of my clients have generated so much data for a phase 1 package using precision cut lung slices, ALI cultures, but in the setting of generating efficacy data really rather than safety data. But they were able to use all of that data, including the in vivo traditional tox studies, to then put it all together and do the maximum recommended starting dose for first in human. And you had so much more confidence then because you'd done all of these other things, which were all more or less giving you the same answer. So, but we're not at a stage where we can say, well, we've done that, so we don't need to do a 14-day and a 28-day. We're definitely not there yet, certainly for pharmaceuticals. Vicky, I don't know if you wanted to add anything.
Yeah, no, really nicely summarised. Yeah, I mean, that was probably a really good case study, detailed that actually we can do a lot of other work that supports that. We don't have to run long 90-day studies. There is a lot of work like that goes into submissions, but it's not necessarily published
Right.
and accessible. So I do see aspects of it. Yeah, I mean, there's a lot of potential, but I think it's really defining what it is and using all those. A lot of those techniques are quite new. They're still quite exploratory, but and having the forum to be able to combine them to show that evidence, that weight of evidence that maybe the studies don't have to be done. But that's definitely emerging as an area we absolutely need to work together on to.
Yeah, we need to get you involved in the work stream as well.
Yes, sure. Sign me up.
Okay.
No, I think the issue is really to find a correlation, because especially when you work with a monoclonal, let's say cancer-based cell line of epithelial origin, is a very simplified method. And you can do acute, I mean, if the cells die, kind of likely the animal will die as well. But of course, in most cases, like you can smoke cigarettes for 40 years and then you develop COPD. How can I replicate that in a simplified model? And I think you can't. I think we have to be mindful that an in vitro model is what it is. It's an extremely simplified tool to answer a very simplified question. And then we have the ex vivo models that are more complex. The precision cut lung slices, the IPLs, they can give you probably more data. And I think in the short and medium term, this is where 3R efforts should probably focus on when it comes to PK assessment and probably some pharmacodynamic and also safety data, because they can be kept in culture, the slices, for a couple of weeks. You can get different readouts, you can look at different markers, but with a cell ALI or not, you can do acute, you can look some mechanistic studies, how is it absorbed, where does it go, but I think that's the end of the flagpole for those.
Goes back to the right model for the answering the right question, doesn't it? There's not a one-size-fits-all model that does everything.
The adverse outcome pathway approach is interesting, but obviously it's not always known what the mechanism is. So if you've got a very detailed one that answers every question, fair enough, but they're not advanced enough yet to use them and answer every question.
And of course, these models can help to develop those AOPs. So.
And when it comes to kinetics, I mean, it's only part of, obviously, the AIT's brief, but in the oral space, CACO2 cells are accepted. Anyone is using them, and the FDA is happy with them, because you pop a tablet pill, whatever you want to call it, and you have a certain concentration in the gut, which you can easily measure, and then you can calculate your kinetics, and off you go to the races. In the lung, we have the patient, we have the inhalation maneuver, we have the formulation, and even comparing data where I insufflate something into an animal that really doesn't want to inhale that moment, and the rat breathes really quickly, and depending on when I push the plunger, I get something in or not. So having a right particle size is probably secondary in that assessment even. So we don't have this gold standard. We don't have the ring studies to different labs even to see how my CACO cell or KALO cell compares to hers. It's a bit frustrating, really.
Yeah. Okay, thanks, Carsten. I truly hate to give you the last word, but I'm afraid we're to our limits of time. So clearly, there's so much more we could have discussed. So you all had grayed out bits of efficacy models you couldn't talk about, other work streams you couldn't talk about, the whole discussion around the different replacement methodologies and how that might look going forward. So clearly, anyone who's interested, join the AIT is the answer there. Thank you very much to all the speakers for staying to time, for covering so much in such a short time, and really keeping us with a very interesting workshop that I think achieved all its objectives, of really just covering that area and giving a flavour of how it all works and how you bring a new medicine through to the point where you can start testing it clinically. So thank you very much.